So thank you very much for giving me the opportunity for the introduction of our activities. Today, my title is Japanese Initiative for the Proposal of OECD Test Guideline TG417 Toxicokinetics. My name is Seiichi Ishida from National Institute of Health Sciences and Sojo University. My research interest is to reconstitute hepatic sinusoid in vitro and construct drug efficacy and safety evaluation system like liver MPS, Lilly evaluation assay, bile excretion evaluation, and mass drug screening system. I used to be the section chief of Division of Pharmacology in National Institute of Health Sciences, and currently I continue my research at Sojo University. I also continue to study in NIHS as a guest researcher. Let me introduce National Institute of Health Sciences, NIHS, first. NIHS is one of the oldest public research institute and leading the regulatory sciences in Japan. NIHS contribute testing, research, and studies towards the proper evaluation of the quality, safety, and efficacy of pharmaceutical products, foods, and numerous chemicals in the living environment. Japanese Center for the Validation of Alternative Methods, JACVAM, belongs to the Center for Biological Safety and Research and leading the regulatory acceptance of alternative test methods in Japan. I'd like to introduce Japanese approach to a proposal of an OECD test guideline. We are discussing to propose that liver MPS for the first pass effective analysis as a context of use of toxicokinetics simulator in chemical risk assessment. I'd like to present background and our initiative for this proposal. We think there are regulatory needs for CIVM, including MPS in OECD test guideline. OECD guidelines for the testing of chemicals are a set of internationally accepted specifications for the testing of chemicals decided on by OECD. One of the reasons is that animal welfare concerns are dealt with by ensuring that animal tests are only permitted where necessary. This means that the new test guideline should be basically in vitro test. This is the list of number of test guidelines on human health that do not use animal testing in OECD at 2022. As you see, many test guidelines are adopted in vitro methods. In vitro methods are more than half to 100% in some evaluation area like skin corrosiveness, skin irritation, phototoxicity, eye irritation, and so on. However, most of them are related to hazard assessment of skin and eyes. Thus, the development of test methods for organs in internal body must be prompted from now on. In this context, the prediction of organ distribution, exposure of chemicals absorbed into the body is essential to predict the risk of exposure for each organ. With the anticipation of in vitro toxicity assessment development of various organs or tissues, future toxicokinetics should be positioned as a means to provide to weight that result of AOP-based hazard assessment in various organs or tissues based on IATA concepts. In other words, toxicokinetics is the key to integrate each alternative hazard assessment method. Based on these considerations, we are focusing on toxicokinetics is one of the MPS COU in regulatory field. Let me explain OECD TG417 toxicokinetics. OECD TG417 is a test guideline that describes in vitro studies that provide information on mass balance, absorption, bioavailability, tissue distribution, metabolism, excretion, and basic toxicokinetic parameters. That is to say, toxicokinetics. Information from toxicokinetic studies helps to relate concentration or dose to the observed toxicity and to understand its mechanism of toxicity. However, the test guidelines are used to be based on animal experiments. Chemical risk assessment is now introducing IATA, integrated approaches to test and assessment, which combine several in vitro tests developed based on AOP key events. Consideration of weight of each test for their combination. It is important to predict the kinetics and the distribution of chemicals in the body and tissues. The prediction of internal exposure under new or inaccessible conditions for chemicals is required, and in silico PBK models are intended to estimate target tissue dose in species and under exposure conditions for which little or no data exists. However, sometimes it is difficult to predict the kinetics of chemical substances in silico model based on their chemical properties. This study is one of such cases, predicting of the absorption of micro and nano plastics related from plastics released from plastics in the environment in the human intestine. In this investigation of the effect of chemical substances on human health by animal tests, occasionally insoluble compounds give pathological findings in organs. In such cases, it is difficult to predict their in vitro behavior without wet experiment. We are focusing to substitute the first part of the toxicokinetics modeling, that is the evaluation of the first pass effect with MPS. Once we evaluate chemical substances uptaken from small intestine and their concentration in systemic circulation, in silico model could calculate their distribution in other organs and body. These are the already available cells and MPS devices. For the small intestinal epithelial cells, FI6, which is derived from the human iPS cell, is available. Cryopreserved hepatocytes are widely used. MPS, like physiomimic from CN Bio or Humimic from tissues, are also commercially available. Several MPS developed in Japan will be in the market soon. So combining these cells and the MPS devices, we could be able to construct the test system for the first pass effect evaluation. An alternative method for TG417 toxicokinetics is being developed in AMED MPS2 project in Japan. Program 1, led by Dr. Ito at the University of Tsukuba, has selected MS plate, which was developed by Dr. Matsunaga at Nagoya City University and Cinco Chemicals as the model for implementation of MPS to the industry. Program 3, which I organize, is responsible for the standardization of the MPS application. And Program 3 is also bridging the standardization of the protocol developed by Program 1 to fit the proposal to OECD Test Guideline. The process of the new the proposal of new toxicokinetics test guideline is now preparing in AMEDO MPS regulatory science RS project. The one of the final objective of MPS RS project is the regulatory acceptance of test methods utilize MPS. In the MPS-RS project, the reproducibility and the predictability of the protocol will be examined with the aim of proposing it to the OECD test guideline in cooperation with AMED MPS2 project. Not only NIHS, but also PMDA and CIVM are participating in this project. This is a step for the test guideline proposal to OECD. We've done the research and development, and now in the course, it's validation study. Next step is the proposal document writing. So we are looking for one who joined this TG proposal. Anyone who is interested in the activity, please feel free to ask us. The email address is here. Thank you for your attention.